my fucking underwear.
Good morning.
Morning.
Good morning, everybody. Yes, please go ahead and type your name into the chat. Thank you. We're still missing a couple of folks, but we'll give them a minute. Hope everybody's doing okay. Hope your classes are going well. All right, so today our schedule includes finishing up the lab exercise about the examination of living organisms using wet mounts and hanging drops. And we're also going to start talking about simple staining, the procedure known as simple staining. We won't finish that today, but we'll finish that up on Friday. Just a housekeeping note. I'm working on your lab homework questions. I've graded about half of them. Those were the ones that you handed in to me Sunday at midnight. Look for those to be completely graded tomorrow. So if you haven't yet gotten your grade for your lab homework, you'll have that tomorrow. And of course, there'll be another homework set of questions to hand in by this Sunday at midnight. You can certainly start working on those now or after today's meeting, but you won't be able to finish them until after Friday when we're all done. All right. So first things first, does anyone have any questions about any of the lecture videos that you've been viewing? The history of microbiology, a little bit of it anyway. The science of microscopy, and the cell, which is one you may not have viewed yet. You know,
I had a question about one of the quizzes. I think it was the microscopy one, but it was one, I forget what question it was, but like it was a true or false, and two of the choices, they were the same thing.
Yeah. Is this Sarah?
Yeah.
Okay. Yeah, Sarah, I actually sent an announcement out today about that. So after class today, just pull up that announcement. Yes, one of your classmates alerted me to that technical error on Blackboard, and it's been fixed. And again, I sent out some information about it for the folks who have already taken the quiz.
Okay, cool.
If you haven't taken that quiz yet about science of microscopy, you have nothing to worry about. The quiz is fixed. But for those of you who've already handed it in, I've made some adjustments.
Okay, thank you.
Yeah, you're welcome. I did want to mention there's one item in that science of microscopy lecture that students sometimes have some difficulty with, and I just wanted to very quickly talk about that just in case there's any confusion about it. And that's this concept of resolving power that microscopes have. Our light microscopes are excellent and they help us look at microbes every day in the laboratory, but they are limited in this concept of resolving power. And what resolving power is about is how far apart two objects have to be underneath a microscope lens in order for us to see them as two different things. How far apart do they have to be for our eyes and that microscope to show them as two different things? Because if you think about it, at very, very high magnifications, which we're using under a microscope, the closer together two objects get, there will come a point where they're going to look like one object to our eyes. And it's the distance that they have to be apart from each other. That's what's called the resolving power. And for a light microscope, that's typically about 200 nanometers. Remember, nanometer is a very, very small unit. um, there are 1,000 nanometers in 1 micrometer, and there are 1,000 micrometers in 1 millimeter. So 1,000,000 nanometers will fit into 1 millimeter, very, very small unit of measure. So two objects have to be at least about 200 nanometers apart from each other on a glass slide in order for the microscope to show them as two separate objects. So we have to be able to use that information a little bit in the sense that, for example, if you were told that two objects were lying 100 nanometers apart from each other, you'd be expected to be able to explain whether they could be viewed as one or two. 100 nanometers falls in that range where the microscope just can't magnify them enough for us to see them as two things. So the take-home message is, if two objects are less than or equal to the resolving power, again, which is typically about 200 nanometers, if they lie less than or equal to that distance from each other, we're not going to see them as two things. It's just going to look like one thing under the microscope. That's what resolving power is about. How good is that microscope? How powerful is that microscope at resolving or separating objects on a slide? So less than or equal to the resolving power, we see them as one thing. Greater than the resolving power, we see them as two things. So again, just something that students have historically struggled a little bit with on examinations and things. All right. The other thing I just wanted to mention very quickly is if you look on your schedule for Monday, you have your very first lecture exam. And we'll talk a little bit more about this on Friday. But your first exam covers the lecture material that we've discussed or presented, I should say, so far in the course. So that's everything up to and including the lecture material this week called The Cell. You're going to have the opportunity to take the lecture exam on Monday at your choice of time. It will be available to you from 8 a.m. in the morning to 8 p.m. in the evening. And you just have to choose a time in between where you can sit down and take the exam. You have 90 minutes to take the exam. We're going to talk a little bit more about the exam on Friday, but I just wanted to give you a heads up so you start thinking about that and make sure it's on your calendar. All right. So what I'm going to do now is jump back in to the lab exercise we started last time. If you recall, we've been talking about wet mounts and hanging drops and why we would want to use those. And I finished up by saying that I created a culture, if you will, in my kitchen by mashing up a strawberry into a little bit of water, because I know that strawberries tend to have a lot of yeast in their microbiome on their external surface. And if you mash them up in a little water and then leave them for about a day, leave that watery material for about a day, you'll grow enough of that yeast to be able to see it under the microscope. So I'm going to go ahead and go back to our slides for this exercise. Now you should be looking at the image of my hand holding a test tube with that reddish liquid in it. Well, what I'll do as I talk through how I made a wet mount here is I'm also going to review for us some of the techniques we've been talking about for a microbiology lab and the safety measures that we use. You'll notice that I have a gloved hand. Anytime we handle culture material, we handle it with a glove. Now, do I think there's anything dangerous in this tube? No. I doubt it very, very highly. But I'm going to treat every single container of culture material as though it contained a dangerous pathogen. That way, if I ever am handling a dangerous pathogen, I'll be safe. So you'll notice that this test tube happens to be plastic. I've shown you a couple of images previously of glass test tubes. We also use plastic test tubes quite a bit in the laboratory. So this is considered a single-use item. This would get disposed of when we're finished. Um, so remember what we said about making a wet mount. We said the very first thing we want to do is we want to make sure that this culture, this volume of fluid that has microbial cells growing in it, that's what we defined a culture as. This collection of yeast cells growing in here. We have to mix this up really well before we draw a sample out of it. So I didn't take a picture of that, but I mixed up that culture very thoroughly, and now I'm ready to take a sample. Remember, microbes are not going to jump up off the counter and get into your sample. They're going to fall out of the air and get into your sample. So we do several things to try to prevent our sample from being contaminated. Number one, when we open the tube, we're going to keep it open for as short a period of time as possible. Number two, we're going to keep it at an angle because that's going to discourage things from falling down and getting into the opening. We're going to hold the cap in our fingers, in our hand, and not put it down on the desktop, because if I put it on the desktop, I could easily contaminate it. Now, the next thing I have to do is put some kind of a sterile device in here to draw up some fluid. I'm going to use a sterile, disposable transfer pipette. Remember that when you put your device into the fluid, you want to draw up a sample from the center of the tube. Don't put your inoculating loop or your swab or your transfer pipette all the way to the bottom. Don't put it just under the surface. Put it right about midway into the tube to pull out your sample. So you can just see in this image, this plastic transfer pipette. Remember, these have a bulb at one end that we use to draw up fluid and then expel fluid. Very, very handy little instrument to have in the lab. Single-use item. They come both sterile and non-sterile. I would want a sterile one because I wouldn't want to accidentally contaminate my sample. I'm placing a nice drop onto my glass slide. Remember, I need to label that slide. Always, always label slides, initials, date, and what is on the slide. So if somebody else picked it up, they would know what it was. Now in this view, you're seeing my hand putting the cover slip onto the slide. I know it's hard to see exactly where this square cover slip is, but I'm outlining it right here. Remember, these are very, very thin, very fragile pieces of glass. And the technique for putting it down on top of your wet mount drop is to put one edge down first. You'll notice that the drop automatically pulls over and along the edge of the cover slip. That's that capillary action that you learned about in physics or chemistry. Once I see that happening, I can slowly lower the other side of the cover slip, and I'm going to avoid too many air bubbles underneath my cover slide. So I have a wet mount now, and it's ready for examination, which is why I've got the microscope out and available now. Notice that my hands are not gloved anymore. Once you have prepared your slide, whether you're making a wet mount or a hanging drop, or if you're staining a slide, which we'll talk about later today, once you go to the microscope, gloves come off. And the reason is because we wouldn't want to contaminate this microscope with our gloved hand. Remember, if you're wearing gloves, you should assume, you should act as though those gloves are contaminated, even if you know they're not. You should behave as though they were contaminated with microbes. So don't touch anything like a microscope that you cannot routinely disinfect. We can certainly wipe our microscope down, and we do periodically, but we cannot disinfect this instrument. Any questions about what I've said so far? About the technique of handling a culture and tilting the test tube and using a sterile instrument to pull out my sample and so on. Any questions about that? Okay, good. Now take a look at this image on this slide. What you're looking at here is a Sharpie line that was drawn on a glass slide. Remember, when we make slides to look at, excuse my dogs, they are policing the neighborhood as usual. When we make a slide to examine microbes, we're going to place our material, whether it's a drop of fluid or some other type of material, we're going to place that right in the center of the glass slide, right in the middle third. And when we're first learning, it's helpful to draw a circle on the slide, literally, with a Sharpie pen or a grease pencil, just to show us where we put our material. You're going to see when we start talking about staining that, I apologize. I apologize, folks. There's a stray dog running around my neighborhood this morning, and my older dog is very unhappy about that. So my point was a moment ago that sometimes it's hard to find cells when we're first learning how to use a microscope. It's hard to find them and focus on them. And the best way to do it is to give ourselves a landmark. And we can see our Sharpie circle really easily. Underneath the microscope, you can see it here. If you can find your line, you can then find the cells nearby. Not everybody uses that technique, but when you're first learning, it's especially helpful. All right, so we are now in focus under our low power 4X lens. Remember, we call all of the lenses on the microscope, we call those objective lenses. We have a low power, we have sort of a medium power, 20X or 10X. Then we have a high dry objective lens, that's the 40X. And then we have an oil immersion lens, that's the 100X. So we're under the low power lens here, 4X lens, which means we have a total magnification of 40X, 40 times, because we multiply the lens strength by the eyepiece magnification, which is always 10 times. Now, when you look here under that low power, you can see a couple things. First of all, you can see this very distinct blue string. And then you can see this brownish looking material that looks almost like branches or a web of material. First things first, this blue string is just debris. It's debris in my sample. I don't know how it got in there, but it's some kind of a fiber. And it happens a lot that when we're using real world samples, we get bits and pieces of things into the sample accidentally. In other words, this was likely either on the strawberry or when I mashed the strawberry up in a bowl, I somehow introduced this blue fiber. Now, the brown material that you're seeing, and of course, it's not stained, remember, this is a wet mount. This is the yeast that I expected to see growing on this strawberry. I want you to notice that it has a real branching pattern that's very typical of fungi under the microscope. This branching pattern, these are what we refer to as hyphae. Hyphae are essentially the branches that mold makes. Now, we haven't talked about fungi yet, and we will later on, but under the microscope, we're going to see two types of fungal structures. We're going to see molds and we're going to see yeast cells. The molds are what you're looking at here in brown, the hyphae, the branching mold. Now I've taken us up under the oil immersion lens, so the 100x lens. So we're now at 1,000 times magnification. Again, no stain on the slide, so everything is basically the same color. And you can see why sometimes it's not easy to examine things without a stain. You can still see the branches, only they're much bigger now. They're much more magnified. These are hyphae. This is fungus. And these round, sort of egg-shaped structures, these are the yeast cells. So I can see a few yeast cells over here out of focus. Here's one that's pretty well focused. Here's one that's really nicely focused. You can see that this cell, this yeast cell, is emerging from the end of one of these hyphae branches. And that's how this particular species of fungus grows. The yeast cells emerge from the mold. The other thing you can see really nicely here is this. This particular yeast cell is dividing and making more yeast cells. This species of yeast, and all yeast, no, I take that back, not all yeast. This species of yeast, though, uses a process of reproduction called budding. When it divides, a new cell comes right off the surface of the old cell. It buds, just like a little flower bud emerges. So this is an asexual type of reproduction. It's not that a male and a female yeast cell are coming together to reproduce. This is one cell that is reproducing on its own, asexually, through this process where the new cell buds right off of the old cell. Very efficient way to reproduce, which is why yeast use it. Now on this next slide here, I've just moved the stage. I used those black knobs on the right side of the microscope that are under the stage to just move the stage and hence the slide, so I can look at a different part of the slide. And that's what I'm seeing here. Again, you can really see these branches, these hyphae in some detail. It almost looks like they have bark on them, doesn't it? These are hyphae. That's the mold. And then there's all kinds of circular egg-shaped yeast cells. Some of them are out of focus and some of them are in focus. When you look at a wet mount, you have to remember that you're looking at several layers of material underneath that cover slip. It feels like it should just be one layer because the cover slip is sort of smashing things down, but you're going to get multiple layers of material. So you have to be ready to focus in and out until you find what you need to see and get it in focus. So take a look at this right here. This is a nice long line of yeast cells that are emerging from this branch, this hyphae. I want you to notice, and I know this is hard to see, but there's a structure right here in the branch, and there's another one harder to see right here. Can everybody see that? Those are actually nuclei. You're looking at the nucleus here and another nucleus here in this fungal hypha. Now, one of the things to remember about fungi is that they are eukaryotic. They are eukaryotes. Remember, there can be microbes that are eukaryotes, and fungi fall into that category. Yeast and molds are microscopic, so that falls into our microbe definition. But only the yeast are single-celled. Only the yeast are single-celled. These are not single-celled. These are multicellular structures, these branches, these hyphae. And that's why you can see two nuclei right here in the same branch. This branch is made out of several different cells. So when it comes to the fungi, both mold and yeast are microscopic, but only yeast are single-celled. So yeast meet our definition for microbes. And the other thing I'll mention, and again, we'll talk about this more when we talk about fungi in detail. You might wonder why these cells are all here. Don't they fall off? Don't they break off? And the answer is yes, they do break off. But it turns out in different species of fungi, it's either easier or harder for them to fall off this branch, this hypha. So it depends on the species. These chains of yeast cells can get quite long. Eventually, these cells will fall and be their own unique organism. But as you can see here, sometimes they hang on for quite a while. Remember what this is, this chain. This was a yeast cell that reproduced through budding and made another cell. And then this cell reproduced and made this cell, and so on and so on and so on. They're growing, they're reproducing. They just haven't fallen off away from their family of cells yet. One more image here to see. Again, I'm just off to the side here. So that's what you're looking at. You're looking at the very edge of the view underneath the microscope. And just to show us again that there's these lovely branching hypha. Yeah, see, I told you I'm a nerd. Branches and branches of the fungal hyphae growing, and we can see all of these yeast cells emerging off of the hyphae. Yeast are an unusual organism. They are very unlike all of the other eukaryotes because they take many different forms. They have complicated life cycles where they start off as one form and then they become another form. They can reproduce in different ways depending on the species. They're really fascinating. Now take a look at this slide. This was, again, on the same slide as what we've been looking at. I just moved to a different area on the slide to take this image. I want you to look here because we can learn an important lesson from this slide. Again, it's a wet mount. Everything is the same color. It can be a little hard to see. But if you look around the slide, there are lots of different things here, and they're all different shapes and sizes. For example, there's something circular right here that's quite dark. There's something nearby that's the same size and shape, but it's a slightly different color. There are these sort of twisted looking bits right here that are a dark color. Here's something quite big and bulky. Here's something else much bigger and bulky. So if you're shown an image, if you look at an image of microbial organisms, something that was taken with the use of a microscope, the very first piece of information that you have to have before you can determine anything else about what might be on this slide is what magnification you're at, because that's going to give you a lot of information. So if I was shown this slide and I didn't know what magnification I was at. I might think, for example, that these little circular things here and here, here's another one right here. I might think that those are bacteria. I might even think that these elongated structures are bacteria. Remember, there are some bacteria that have a rod shape, an elongated shape. So I would be forgiven, frankly, if I looked at this and I said, oh, yeah, these might be bacteria. I would be forgiven until I was told that this picture was taken underneath the 4X lens, the low power lens. These cannot be bacterial cells. These circular cells cannot be bacteria. These elongated structures here, they can't be bacteria. Why? Because I can't see bacteria under the 4X objective lens. That's a total magnification of only 40 times. I need to be under, at minimum, the 40X objective lens, the high dry objective lens to see bacteria. And I can see them best using the 100X objective lens, the oil immersion lens. So you always need to know what magnification you're at if you're going to take an educated guess about what you're looking at on the slide. And of course, regardless of the magnification, even if I am under the 100X objective lens, the oil immersion lens, 1,000 times magnified, even if I am under that lens, I won't see viral particles. I will not see viral particles under a light microscope. Can't happen. You need an electron microscope to see viral particles. So the question then becomes, what am I looking at? If these aren't bacteria, remember, archaea are approximately the same size as bacteria and approximately the same different types of shapes as bacteria. So what the heck are these? These are yeast cells, the round ones, and little pieces of fungal hyphae. Those big, broad, branching trees, not only do the yeast cells fall off individually, but bits of the branches fall off too. And that's what I'm seeing here. This is a piece of mold. This is a piece of the fungal hyphae. Here's another one. Here's another one. This is just a bigger piece. And these little tiny looking circles, these are those yeast cells that we looked at a minute ago. It's just that under the 4X lens, they're very small, they're tiny. You can see eukaryotic cells under the low power lens. They're not very big typically, but you'll see them. You cannot see prokaryotic cells. And remember, there are two kinds of prokaryotes, bacteria and archaea. You won't see those under the low power lens. On this slide, you're looking at the wet mount, the same wet mount, under the low power 4X lens, but I've done something to it here. What I did was I took a drop of stain and I put it right at the edge of the cover slip. Remember, I laid the cover slip down over my drop, my sample drop. So there's a technique we can use with wet mounts where we can add a little stain in. You take a drop of your stain, and this stain happens to be called methylene blue, and you put the drop right at the edge of the cover slip. And what's going to happen is it's going to get pulled, it's going to get sucked underneath the cover slip by capillary action. It's not the same as staining a slide, as you'll see later on. It's not the same technique. It's still a wet mount, but it's a wet mount where a little bit of stain has been added to all that fluid, hopefully to help me see things a little clearer. Now, part of the reason I took this particular picture under the 4X lens is for these white structures. This and this and this and this. These are nice, symmetrical, circular structures. You would be forgiven if you thought that those were cellular, but they are air bubbles. And this is what air bubbles look like under a microscope slide. Very symmetrical with a thick black line around them. And they're all different sizes. They're all the same shape. They're all circular, but they're all different sizes. That's all this is. The reason you can see them so nicely on this slide is because, of course, they're not going to be blue because they're made out of air. It's a bubble of air. I've made all the liquid turn blue by putting that drop of stain into the mix, but the air will stay white or colorless. So you can really see air bubbles well on a wet mount where you've added a little stain. Now take a look at some of the fungus. Again, this is the same slide we looked at earlier. I've just put a little stain in here. We're under the 4X lens in this view. It's not well focused here. So if it looks fuzzy to you, it is fuzzy. We're under the low power lens, 4X, 4 times magnification. We have a total magnification of 40X, 4 times 10. And here is one of those big branching fungal structures. Now, if you look over here, I've gone up to the next power objective lens. I'm not as high as I was earlier. I'm under either the 10X or the 20X, depending on the scope I'm using. But look at how more well-defined the structures are with this drop of stain in here. When we saw these yeast structures without stain, remember they looked like this, you can see them. Of course you can. But some people prefer the contrast that you get when you look with a little bit of dye. Each individual yeast cell is very clearly visible. All right. Who has questions, if any? So wet mounts, they're perfectly good for looking for motility, but they're also just good for looking at living cellular organisms. They're easy, they're quick. It's easy to take, for example, a drop of pond water, put it on a slide, put a cover slip over it, and I can examine it immediately under the scope. And if I can't quite see things the way I want, I can add a drop of stain. That'll get pulled into the water sample. That'll help a little bit. It's very quick. But what we're going to talk about next is how to make a proper stained slide. And you're going to see that the reason people take the time and the trouble to make a stained slide is because you get much better color and much better detail. Any questions at all about what you saw? Again, we're going to talk about fungus later in the course in more detail, but you got to see some today. You got to see some yeast and some hyphae today from our strawberry. All right, so I'm just going to switch gears now. And I'm going to pull up the next set of slides. So you should be seeing a slide that says Lab Simple Staining. Again, all of our lab slides are available to you on Blackboard. You just go to the week's content folder and you'll find a folder that says slides, I think. And then inside there, you'll find both lecture slides and lab slides. So we have a few objectives as we talk about simple staining. First of all, we need to learn the staining process. And then in the second part, what we'll do is we'll actually prepare a smear, as it's called. We will do a procedure called heat fixing. And then we'll use what's called a simple, direct staining procedure. And I'm going to explain to you what those words mean today. Finally, and likely not today, on Friday, we'll look at some stained slides, and we'll get some practice looking at different shapes of bacteria as well. So we have some new vocabulary to learn with regards to staining. The first term is this term smear. When we use the term smear in microbiology, what we're talking about is a very thin layer of cells that have been placed onto a microscope slide. It's critical when we make smears that we get a nice thin layer, because when we go under the microscope, it's going to be very hard to see anything if we have a big thick blob of material. So part of learning how to make a smear is learning how to make a nice thin one. The second term we need to know is this term fixation. When we talk about fixing cells to a slide, or the other thing you'll hear people say is, I fixed that slide. This is a process that will adhere cells to the slide. Now, it also happens to kill the cells, which is actually a nice benefit. We're going to actually kill them as we heat fix them, so there's no more risk to us if those cells happen to be pathogenic. Now, there's two ways to fix cells to a glass slide. You can do it chemically, and you can do it with heat. Now, I've done both in my career. Different labs tend to prefer one method or the other. I personally prefer heat fixation. I just think it works better. But if you do chemical fixation, all you do is you add 95% methanol to the slide. You put a drop or two of a 95% methanol solution on top of the smear, and you let it sit until it dries. Now, methanol is just a type of alcohol. You may have heard of it before. It's particularly toxic, so no surprise that it kills the cells when they get exposed to it. So you put 1 or 2 drops of methanol on top of the smear, on top of that thin layer of cells, and you let that alcohol sit until it dries. Now, the other method uses heat. Now, what's happening with the heat? That's what the mechanism is here. What's happening to those cells is that the proteins that are in the membrane of the cell are going to get sticky when they get hot, when they get heated. Some of the proteins are actually enzymes. And remember, all an enzyme is a protein that is able to act as a catalyst. If you remember from chemistry, catalysts are molecules that help speed up chemical reactions. So cells, living cells, are loaded with enzymes, including embedded in the membrane. So when those enzymes get coagulated along with other proteins in the membrane, when they get coagulated, they don't function anymore. And that's actually a good thing. And the reason it's a good thing is because I told you a minute ago that fixation and staining are going to kill cells. And the normal process after death is for the cell to fall apart, to degrade. By getting rid of some of those enzymes through the heating process, we're going to stop that degradation process. We actually give that process a name. Don't worry about this term too much, but this word autolysis, the auto here means self, and the lysis means break apart. So autolysis, that's the natural process where cells break apart after they die. We're going to prevent autolysis through heat fixation. And we're going to make those cells sticky, so they are literally going to adhere to the glass slide. Now, how do we do it? Well, you can do it with an open flame like a Bunsen burner, or you can do it using a hot surface. And we do have a nice hot surface available to us in most microbiology labs, that device called an incinerator. When you heat up an incinerator, the interior of the incinerator gets so hot that it will sterilize your inoculating loop for you. But it has a metal coating, a metal shell around it. I guess you could call it the housing of the incinerator. And that metal, while it doesn't get as hot as the interior, it does get hot, and it gets hot enough to do heat fixing without destroying the cells. So all we have to do is carefully hold our smear against that hot metal, and we will heat fix the cells. You want to do it for about 20 seconds, and I would say no more than 30 seconds. Remember, heat will fix cells, but if you use heat for too long, or if you use heat at too high of a temperature, you're just going to cook the cells. You're going to destroy them. If I'm going to use an open flame, all I have to do is pass the slide through the flame 3 or 4 times. And we'll see how, I'll show you a demonstration of how this is done. Now, there's a note at the bottom of this slide. I want you to put a star next to this in your lab notes. Only fix dry smears. When we make a smear, depending on our starting material, we're going to get a wet smear. We're using a wet material most of the time. I would say all of the time it's at least a little bit wet, and sometimes it's very wet. After you make your smear, you must let that smear dry before you fix it. And here's why. Remember, we treat every single culture or sample material as though it contained pathogens. If I were to put a wet smear over a flame or on top of my incinerator, what could happen is I could boil the water and send steam up into the air. That will also send cells up into the air. In other words, I will vaporize the wet material on the slide, and I will send potentially pathogenic organisms up into the air in what we would call an aerosol. And if there happen to be dangerous microbes in that aerosol, I have now created a situation where people might breathe them in. So we always make sure that our smears are bone dry before we fix them. On this slide down at the bottom, here's our classic Bunsen burner. You can see the person has it set up very nicely. If you remember from chemistry, you want a nice blue flame here. You don't want a bright orange flame. That's not an efficient use of heat. You'll notice that this person has a pair of hemostats in their hand so that their hand doesn't need to get close to the flame. And they're holding the glass slide. The smear is right in the center of the slide. Now, this picture is a little deceiving because it looks like they're just holding the slide over the flame, like they're cooking a marshmallow or something. You would never do this. You would never put a slide on top of the flame and just sit there. But what you would do is pass the slide back and forth through the flame 3 or 4 times. That's all it takes to denature the protein enough to get it to stick to the slide. The other thing I want to show you is up on this top image. You'll notice there's a little timer set here, and there's what looks like a black hot plate here, and that's exactly what it is. It's a laboratory version of a hot plate. It's called a slide warmer. If you make a smear and it's particularly wet, you can just wait for it to dry, or you can place it on one of these slide warmers. They're not hot per se, but they're hot enough so that they will help these slides dry quicker. It's not so hot that it's going to destroy the cells, it's going to cook the cells, but it's hot enough that it will help them dry quicker. So that tool is called a slide warmer. And you will find a slide warmer in most microbiology labs. All right, so anytime we're making a slide for staining, we're going to go through the process of making the smear, smearing a nice thin layer of cells into the center of our slide. And then we're going to fix our smear using methanol or using heat. And then we do whatever staining process we're going to do. Note that if you forget to fix your slide, if you make a smear and then you forget to do the fixation step and you just go straight to the staining step, what's going to happen is you're going to wash the cells right off of the slide. So having cells rubbed onto a slide, placed onto a slide, and then dry, that's not enough. You have to fix them to the slide. You have to attach them to the slide, or they're simply going to wash away when you do your staining procedure. Now, there are many, many, many staining procedures, and we use them for different reasons in the microbiology lab. But there are some things they all have in common, and that's what we're going to talk about next, some more vocabulary for us. These microbiology stains are just dyes, but they're a particular special type of dye that are made out of salts. Now, if you remember from chemistry, all a salt is a molecule that's composed from two ions. And remember, all an ion is a charged atom. So, for example, when you say the word salt to most people, they think about table salt, which is sodium chloride. Sodium chloride is composed of two ions. It's composed of sodium, which has a positive charge, and chloride, which has a negative charge. When they come together, the charges cancel each other out, and you get this neutral molecule called sodium chloride. There are many salts. Think about, for example, sticking with chloride, think about calcium chloride or magnesium chloride. There are lots and lots of different salts, and we use these salts, some of them, to make these various stains. Now, what's the difference between just a regular salt and a microbiology stain is that the salts that we use for stains have a natural color to them. They have a pigment, a shade associated with the ions. So we call a colored ion a chromophore. Anytime you see this term chromo in a word, that means color. So a chromophore is a colored ion. It's an ion that has a natural color associated with it. Now, here comes some important vocabulary. These are all ions, so some of them have a positive charge and some of them have a negative charge. Positive chromophores, in other words, positive colored ions, are known as basic stains, and negative chromophores are known as acidic stains. So that's terminology that we use all the time. We talk about stains as either being basic or acidic. And we're going to use those two different types of stains in different circumstances. Most of the stains that we use for bacteria are basic stains. Let me say that again, because that's really important. Most of the stains that we use in the microbiology lab, because we stain a lot of bacteria, most of them are basic stains. So they contain a positive chromophore, a positive colored ion. And what happens is those basic stains are going to permeate through the cell wall. Remember the peptidoglycan cell wall? They're going to permeate, they're going to travel right through that peptidoglycan, and they're going to bind to anything that has a negative charge. and the membrane is full of negative charges. So they will bind to the membrane, and they'll even bind to some things that are in the cell itself, depending on the stain. Just as a reminder from chemistry, again, as a review, when we call something acidic, we're saying that it has a pH less than 7. Remember, we consider 7 to be a neutral pH. Anything above 7 is considered basic. Anything below 7 is considered acidic. All right, so now that we have some basic terminology about what's in our stains, we're going to talk about some different protocols of staining, some very general terms that we use to describe different protocols or different procedures for staining. The way we break them down is into either simple staining or differential staining. If a procedure that you're doing is known as a simple staining procedure, what that means is you're only going to be using one stain. And when you apply that stain to your smear, you're only going to get one color visible when you look under the microscope. When you do a differential staining procedure, you're going to be using multiple stains. So you'll put one on and then you'll wash that off, and then you'll put another on and you'll wash that off. Sometimes differential staining procedures involve many stains. You have to keep putting one on, take it off, put the next one on, take it off, put the next one on, take it off. Differential procedures can be pretty complex. Now, when you're doing a simple staining procedure, we can further classify those as being either direct or negative. In other words, there are simple direct stains or staining procedures, and there are simple negative staining procedures. If something is called a simple direct procedure, it means it's going to stain the cells themselves on the slide. If it's a simple negative staining procedure, it means it's not going to stain the cells. It's going to stain the background so that you'll be able to see the cells better. Most of the simple staining procedures that we do in the microbiology lab are direct. Most of the time, we are trying to stain the cells themselves. But there are a couple of very popular negative staining procedures. Now, I've got an example for you listed on the slide. Remember when we were doing the wet mounts, I said I added a drop of methylene blue. Methylene blue is a very common stain in the microbiology lab. It's very practical. It's used for a lot of different things. Methylene blue contains a positive chromophore, so it's a basic stain. It stains cells, not the background of the slide, but the cells themselves. So we would call it a simple direct stain. We can also call it a basic stain. Questions? Questions about staining? One of the most frustrating things when you work in a microbiology lab, if you end up as a technician in a microbiology lab or perhaps doing research in a lab, one of the frustrating things is that stains have a limited shelf life. So if you're the person whose job it is to sort of manage the inventory in the lab, you have to pay close attention to when the stains expire because they won't function correctly after they expire. So luckily for us, most of them last for a good period of time, at least a year, in other words. But there are some stains that are very, very fickle, and they're only good for, a few months after you buy them. So, if you're working in a busy lab and you're going through lots and lots of stains, that's not really a problem. But in some labs, the stains sit on the shelves and they expire, and somebody has to be paying attention to that so that the old expired stains get disposed of properly, and a new fresh batch can be bought. All right, I'm going to dive back in. The next thing that we're going to be talking about, the next thing that we're going to do in this exercise is we're going to watch a video clip together, but we don't have enough time today to watch the video. So I'm just going to say a couple more things. And then when we get together on Friday, we'll pick up here. I want you to notice again, we'll try to review things that we talked about so far as we go through each lab exercise. What you're looking at here on the countertop is a bottle of ethanol, and I use this bottle here at home to disinfect my work surface. I think I mentioned to you that I prefer alcohol over bleach as a disinfectant, only because in my hands, I always manage to get the bleach on my clothing, and I always manage to destroy my clothing with it. The alcohol is much more forgiving to clothing. Remember, you always want to have 60 to 90% solution if you're using the alcohol as a disinfectant. In most labs, it's going to be a 70% solution. That just happens to be how most labs mix it up. If you were to use a 100% alcohol solution as a disinfectant, it just wouldn't work as well. It's one of those things that seems counterintuitive. You would think that a more concentrated solution would work better, but it turns out you need a little bit of dilution to get the best effect. Now, the other thing I want to call your attention to is the warning label on this bottle. This is a bottle that you would buy commercially. And you can see it's got this curved spout on it, which is why people like it, because you can pick this bottle up and you can easily squirt the alcohol down onto the counter. So people purchase these through chemical supply companies. They're found all over different types of laboratories. And here is that diamond that we talked about in the biosafety laboratory exercise. Remember, there are going to be 4 boxes. Each one is a different color, and each color refers to different things. And remember, we said the numbers that you're going to see range from 0 to 4. You will see a number in blue, red, and yellow, but you may not see a number in the white box. If you remember, and you may not, but if you remember, the white box was for any special hazards. So if there's no number in the white box, that just means there's no special hazards in this bottle. What do I mean by special hazards? Well, for example, there are some things that can spontaneously ignite when they are exposed to the air. So that kind of thing would have a number down here. But this is just ethanol, and it has no special hazards, but it does have some other hazards, doesn't it? There is a fire hazard that's relatively high. Alcohol is quite flammable. And there's a health hazard. Now, this one's a little bit lower. This one's only a 2. But you probably are already aware of the fact that ethanol has some health hazards associated with it if you consume it or get exposed to it over a long period of time. So just a nice little review there of some biosafety. All chemical containers inside a working laboratory should have this information on them, but most of the time it's in very small print. It's your responsibility as the person who is working in the lab to familiarize yourself with the degree of hazard for the materials that you're handling. That falls on you as an employee. All right, that's all we really have time for today. So I'm going to stop here. And if anybody has any questions, we can certainly talk about them. Like I said earlier today, now that we've gone through the examining living organisms material, the wet mount and hanging drop material, there are a couple of questions on the homework that you can already answer. You won't be able to do all of them until Friday, but if you want to start that homework, you'll certainly be able to do that. Right. Does anybody have any other questions or problems? Anybody? All right, good. All right, very good, folks. I'm going to let you go. I will see you again on Friday morning. Keep up with your lecture material.
Thank you.
Okay, we will see you next time. Have a good rest of the day.
Thank you.
Thank you.
Have a good day.
Thank you.
Thank you.
You're welcome. We'll see you next time.
All right, bye.
Should I wait for Sean to log off?
Do you need any help with anything, Sean? I'm going to meet with Nevada for just a minute. Sean, are you still with us? He may have stepped away.
Okay.
I'm going to go ahead and close out for him. So what can I help you with?
I guess I'm just struggling a little bit with some of I guess keeping up with the lectures and stuff, and I'd wanted to ask also just I know we turn our cameras off during class and everything, but I know for me at least, in my other classes, it helps to keep it on just to kind of hold me accountable.
Oh, sure.
For
No.
paying attention.
Absolutely. And you know what? There's nothing wrong with that. Okay. You feel free to do that.
Okay, I just wanted to make sure. And
how many courses are you taking, Nevada?
I'm taking this class, calculus, physics, the New Englander.
Yep.
And research thesis.
Okay.
But calculus conflicts with this time for micro, so I kind of have to work remotely for well, everything's remote, but I guess I have to go back and rewatch those lectures for calculus as well.
Okay.
So
you have a busy schedule.
Yeah.
you have a load.
Yeah.
I've said it before and I'll say it again. The hardest part about taking online classes, especially when the lectures are asynchronous, is staying up with the lecture material, because you don't, when you're in person, you go to a room and you sit down. I mean, you're kind of forced, right?
Yeah.
One of the tricks that I know some of my students have used that they say is helpful when you can is to just set aside the time each week to do it. Like maybe the time that you had originally scheduled your lab for this course. You could say, okay, on Monday afternoon, and I'm just making this up, but at Monday afternoon at 2:00, I'm going to do the first lecture.
Okay.
And on Wednesday afternoon at 2:00, I'm going to do the second lecture. And you just try to do that with your courses. Set a set time. And the other thing that's really helpful, if you can, is to set aside a place where you watch them. Now, I know it sounds silly, but there's actually some real data that shows that if we're sitting in a place that is devoted to work, we will be better apt to do the work. We'll be more likely to do it. So a lot of people, when they're learning remotely, they might sit on their bed or they might sit on the couch. And there's nothing wrong with that. But you'll find that if you make yourself like go to your desk at 2 o'clock and watch the lecture, if you put it on your schedule, you're more likely to do it, and you're more likely to stay on schedule. I know it sounds weird, but it actually works. It's
Okay.
too easy for us when we're not super structured like that. It's too easy for us to say, I'll do this later. I'll do this tonight. And then you get distracted by something else, and you get pulled away. Treat it just like you were going to go to the lecture hall for an in-person lecture. Treat it like that. Say, I'm going to my desk because it's 2 o'clock and I'm going to watch this lecture. And you may find that a little bit more helpful. It's very easy to get behind in an online course. Super, super easy.
Yeah.
I've taken online courses for like continuing education for things. And it's just super tempting to, let some work slide. So
Mhm.
you're not alone in that, believe me. But yeah, you might want to try that.
Okay.
How about when you watch the lectures and you take notes and things, and then you take the quizzes, how are you finding the quizzes?
The two quizzes I've taken so far, I think it was for the microbes one, our first one, and then the
Yep.
lab safety.
Yep.
I found those to be pretty straightforward, and like
Okay,
I
good.
took good notes,
Good.
and so I felt like those were okay.
Okay.
I think it's just like kind of getting into like a routine of trying to make time for like the watching the lecture videos and make sure I am taking the thorough notes.
Yeah. And the other just sort of basic thing that I've heard, you have to figure out when, and this varies a lot from person to person, but you have to figure out what time during the day that you're most like awake and aware and able to keep your attention. There are a lot of people that, are morning people and they watch their lectures at 9 a.m. or whatever. But there are other people who prefer to do it at night because they just feel like they get more out of it at night. That's another trick that people use. You just try to figure out where you're most aware and awake, and you can get the most out of it. Something
Okay.
else to think about too. Yeah.
Okay.
So what's going to happen on Monday is we have our first lecture exam. So between now and Monday, you're just going to want to make sure that the notes you have for those 3 lectures, the intro one, and then the history and the cell one, that your notes are in good shape. If you have your notes in good shape, you're going to be fine during the exam because you can have your notes right next to you during the exam.
Okay.
Okay. And it sounds like your notes are already in pretty good shape if you're able to do the quizzes without too much difficulty.
Okay.
But that's what I would recommend trying. You've got a challenge this semester because you've got that heavy load of classes. And you've got to figure out how you're going to order the time each week to do the work for those 4 classes. Yeah. And again, it really does help to just schedule it. Just put it on a piece of paper and hold yourself to that schedule. And if something comes up and your friends say, "Hey, let's go out to lunch," or whatever, you have to say, "I can't go out to lunch because at 1 o'clock I'm going to watch my calculus video," or whatever. And just try to be strict with yourself that way.
Okay.
Anything else?
No, I think that was it. I think that's helpful for me to kind of think about and do.
Again, just remind yourself from time to time that you are not alone in that struggle. You are not even close to being alone. I don't know very many people who find it easy.
Yeah.
In fact, I can't think of anybody I know that finds it easy. It's very challenging to hold yourself to a schedule when you are at your own place, at home, on your own time. It's
Yeah.
much easier to go to a lecture hall in person.
Mhm.
Yeah. So
Yeah. I
um,
think this year has been such like a learning curve because I'm a senior this year.
yeah.
And so like the last 3 years I've gotten into this routine of getting used to like how I learn and like going to
Yeah.
lecture halls and everything. And then now everything is like you kind of have to relearn how you learn.
And you're really challenging yourself to develop this new skill or skills, because it's also a skill to be able to just not be with the lecturer, but
Yeah.
to be watching and taking your notes. That's a whole different skill than being in the room with the person.
Mhm.
Yeah. I hope that in the long run, when all of this is over, I hope that students will look back and be glad that they were challenged this way. But I know that while you're in the middle of it, it's just hard. It's just hard. The only other thing that just occurred to me that you might want to think about, if you know someone else in the class, um, you could sort of buddy up and try to hold each other accountable.
Okay.
You know what I mean? So
Yeah.
if you're supposed to watch the lecture on Tuesday afternoon, your friend could check in with you and say, did you watch it? And if you didn't, they can fuss at you a little bit, right? Sometimes it's
Mhm.
helpful to have sort of a partner to go through it together. And you could maybe share notes with each other too, you know? So something else to think about.
Okay.
All right. Well, let me know how it goes. And if you start to fall behind, or, you start to have any more difficulties beyond this, please let me know and we'll try to figure out solutions for you.
Okay.
Okay.
Thank you.
You're going to get through it. I know you
Thank
are.
you.
But I don't envy you doing calculus and physics at the same
No.
time. That's a challenge. All right, Nevada, if there's
Okay.
nothing else, I'll let you go.
Okay, thank you.
Okay, enjoy the rest of the day.
You too. Bye-bye.
Bye-bye.